Scientific breakthroughs often start with a simple question: what is? For years, scientists noticed odd, repeating sequences in bacterial DNA, like a refrain in a song with unique phrases in between. These were called clustered regularly interspaced short palindromic repeats, or CRISPR. At first, no one knew their purpose. They were just a genetic mystery, but patterns in biology usually mean something important. Francisco Mojica and others discovered that the unique sequences matched the DNA of viruses that attack bacteria. It was as if bacteria kept a genetic most wanted list of their viral enemies. This wasn't junk DNA, it was a defense system. Bacteria were storing snippets of viral DNA in their own genome, creating a genetic memory bank. If the same virus attacked again, the bacterium could recognize and fight it off instantly. This was a primitive immune system hidden in plain sight. The discovery turned CRISPR from a curiosity into a hot research topic. Scientists soon found special proteins, CRISPR-associated or CAs proteins, linked to these repeats. These CAs proteins acted as the system's enforcers, using the CRISPR library to target invaders. The stage was set to uncover how this system actually worked. What began as a strange pattern in DNA was about to become the foundation for a revolutionary tool. Nature had already invented the most precise gene editing system we'd ever seen. The world of genetics would never be the same. So how does this bacterial defense system work? CRISPR is a two-part toolkit, a guide RNA and a protein called Cas9. The guide RNA is built from the viral mugshots stored in the bacterium's DNA. Its job? To find a matching sequence of DNA, like a search dog trained for one scent. The Cas9 protein is the muscle, a pair of molecular scissors. But Cas9 only acts when the guide RNA finds its target. Together, they patrol the cell, scanning for viral DNA. When the guide RNA locks onto a match, Cas9 cuts the DNA with surgical precision. This snip destroys the virus's genetic instructions, neutralizing the threat. It's a simple, elegant defense perfected by evolution. The real breakthrough came when scientists realized they could reprogram this system. By designing custom guide RNAs, we could target any gene in any organism, pair it with Cas9, and you have a programmable gene editing tool. Suddenly we could cut and modify DNA wherever we wanted. What started as a bacterial survival trick became a universal tool for genetic engineering. The possibilities were staggering. We could edit plants, animals, even humans. CRISPR was no longer just a curiosity, it was a revolution in the making. The age of precise, affordable gene editing had arrived. CRISPR's transformation from curiosity to tool hinged on a few key moments. In 2012, my team and Emmanuel Charpentier's group fused the RNA components into a single, easy-to-use guide. This made the system simple and powerful. We showed it could cut any DNA at a chosen spot in a test tube. Soon after, labs like Feng Zhang's proved CRISPR worked inside living human and mouse cells. Suddenly, editing the genes of complex organisms was possible. CRISPR was faster, cheaper, and more accurate than anything before. Labs worldwide adopted it overnight, accelerating discoveries in medicine, agriculture, and basic biology. New CRISPR systems and improved editors soon followed, expanding what was possible. The gene editing revolution was officially underway. With CRISPR's power came urgent ethical questions. Somatic editing, changing genes in body cells to treat disease, was widely accepted. But germline editing, altering DNA in embryos or reproductive cells, was far more controversial. Changes here would be passed to future generations, potentially altering the human gene pool forever. The idea of designer babies raised fears of new inequalities and genetic divides. Who gets access? Who decides what's acceptable? The risks are real off-target effects, unintended consequences, and the possibility of introducing new diseases. Most scientists called for a moratorium on clinical germline editing until we understand the risks and build a global consensus. We must ensure that technology benefits everyone, not just a privileged few. The stakes are enormous. CRISPR gives us the power to rewrite life, but we need wisdom, caution, and strong ethical guardrails. The science is moving fast, our ethical frameworks must keep up. The future of gene editing depends on the choices we make now. While debates continue, CRISPR is already changing lives. In medicine, it's being used to treat genetic diseases like sickle cell anemia. Scientists edit a patient's stem cells, correct the faulty gene, and return the healthy cells. Early results are life-changing. CRISPR is also supercharging cancer immunotherapy, making T-cells better at fighting tumors. In agriculture, CRISPR is creating crops that are more nutritious, resilient, and longer-lasting. Imagine mushrooms that don't brown, or tomatoes with higher yields. This could help address food security and reduce waste. In research, CRISPR lets scientists quickly turn genes on or off to study diseases from Alzheimer's to heart disease. It's even powering rapid, affordable diagnostic tests for viruses like COVID-19. CRISPR is no longer just a theory, it's a tool reshaping medicine, food, and science itself. The impact is real and it's only just beginning. The future is already here. In 2018, CRISPR's ethical dilemmas became reality. A Chinese scientist, He Jiankui, announced he'd edited the genomes of twin girls to resist HIV. The global response was outrage. His experiment was secretive, reckless, and unnecessary. He exposed the children to unknown risks, violating scientific and ethical norms. The edits were incomplete and unpredictable, showing the science wasn't ready. He was condemned and imprisoned, but the damage was done. The incident was a wake-up call. Self-imposed guidelines weren't enough. We need enforceable rules and global oversight for human gene editing. The He Jiankui affair stands as a stark warning. Ambition must never outpace responsibility. CRISPR's promise is immense, but so is our duty to use it wisely. The world is watching and learning. The future of CRISPR is dazzling and complex. Soon, we'll see CRISPR-based therapies for more diseases, from blood disorders to muscular dystrophy. Delivering CRISPR directly into the body is the next big challenge. New tools like base and prime editing will allow even more precise genetic fixes. Scientists are exploring ways to turn genes on or off without permanent changes. In agriculture, CRISPR will help crops adapt to climate change and reduce environmental impact. In conservation, gene drives could control pests or fight disease, but carry ecological risks. The technology's reach will extend far beyond the lab. Its future depends on global conversations among scientists, policymakers, and the public. We must build strong ethical frameworks to guide its use. CRISPR lets us rewrite the code of life. What story will we choose to tell? The next chapter is up to all of us.